Welcome to Module 15, where we are going to be going over information from Chapter 17. So this chapter is going to focus on cell reproduction and cell differentiation. This chapter will actually go from Module 15 into Module 16 as well. So it's basically going to take up 2 modules. Both video sets or the videos will be posted in both modules, so it'll kind of be the same information. It's just that I'm giving you 2 days to go over this information because it is a little more detailed and a little more nuanced than some of the stuff that we've covered in the previous chapters. So with cell reproduction, we're going to talk about how cells divide the DNA and how the DNA is replicated. And then we'll also talk about how DNA translates or DNA is translated into RNA and then transcribed into proteins. And then for the last part of the chapter, we'll focus on mitosis and meiosis, which is how cells divide and replicate themselves. All right, so to get started, right? So cells reproduce basically by dividing themselves in two. So we talked about unicellular organisms and multicellular organisms earlier on in the semester. Unicellular organisms, so like your prokaryotes, they reproduce through cell division. So cell division is basically the only way they can reproduce. Multicellular organisms reproduce either asexually or sexually, but their cells divide throughout their life, and that enables them to grow from a embryo or a fertilized egg into a multicellular individual. And so we'll talk a little bit about those differences as we kind of go through this chapter. The mechanism for cell division is exactly the same in all eukaryotes. Okay, so the cell cycle is kind of how or the cycle in which cell division takes place. And so what we first need to do is we first need to go over the basics of the cell cycle, and then we'll talk about kind of what each phase is or each part of the cell cycle is. So the cell cycle includes 2 major phases. One is interphase and one is the mitotic phase or the meiosis phase, depending on which cell division process you're talking about. So interphase Okay, so I had something happen. I have absolutely no idea what it was, but okay. So interphase is the cell phase that is between the cell division process. And this is when the cell grows and prepares itself for the division process. And it also prepares the DNA for the cell division process by replicating the DNA inside the cell. And so interphase is made up of 3 different phases called the G1 phase, the S phase, and the G2 phase. G1 basically stands for first gap, G2 stands for second gap, and S phase stands for the synthesis phase. So G1 is basically the primary growth phase. This is when the cell is very active. It's basically doing cell things, cellular processes, right? Cellular respiration, all of that good stuff. After the first growth phase, the synthesis phase then starts. And the synthesis phase is when the DNA replicates itself and gets ready for cellular division, so you can have 2 copies of your DNA put into 2 daughter cells. Now, the G2 phase, which is the second gap phase, is the final growth phase before cell division happens. So any kind of final growing that the cell needs to do, this happens in the second gap phase. Now, when interphase finishes, the cell will go into the division phase, which is known as the mitotic phase or the meiosis phase, depending on, again, the division process. For the mitotic phase, the cell will undergo mitosis. This is basically where the nucleus will be divided, the duplicated DNA is distributed evenly into the two daughter cells. The nucleus divides, and then cytokinesis happens. Cytokinesis is a division of the cytoplasm, which ultimately forms two new daughter cells. And so at the end of mitosis, you have two daughter cells that are genetically identical, so they are exactly the same. In general, the complete cell cycle takes around 18 to 24 hours. Mitosis and cytokinesis, though, takes less than an hour to complete the cycle. So the whole division process is actually about an hour long, but the whole cell cycle is about 18 to 24 hours. Some cells can also enter what's called a non-dividing state, so G sub 0, and then they can do this either temporarily or permanently. So for example, after we finish growing after adolescence, our neurons and our bone cells, so our osteocytes, will enter that non-dividing state, and they will not divide anymore after that. So here's an image showing the cell division, or the cell cycle, with this kind of, with the outer line here, right, this area being interphase. So these three yellow triangles are the three phases of interphase. So G1, S phase, and G2. And then this triangle here is the mitotic phase. So this is when the nucleus, or I should say this is when mitosis happens, the nucleus divides, and then cytokinesis will happen, which divides the cell. Now, looking at the DNA structure, in order to understand how the DNA is separated in mitosis and meiosis, we first need to really understand how the DNA is structured and what happens with the DNA once it's replicated and then separated in those two different cell division processes. So the human DNA is organized into 46 different chromosomes. And those chromosomes contain about 3 billion base pairs of DNA. Remember, the base pairs are the adenines, the guanines, the thymines, and the cytosines. And so on these 46 chromosomes, the 46 chromosomes are basically two pairs or 23 chromosomes that are made up of two pairs, okay? So for example, you have two copies of your first chromosome, your second chromosome, your third chromosome. And so humans have 23 individual chromosomes, two copies each, making 46 separate chromosomes. Now, these chromosomes are made up of DNA that is wound around proteins that are known as histones. And then they will condense down and create the chromosomes. When the DNA is loosely configured in the nucleus, it's referred to as chromatin, right? So it's not until it's compacted into chromosomes during the mitosis phase or before the mitosis phase, prophase is when that happens, that they're visible underneath the microscope. And the replicated chromosomes, so the two sister chromatids, will be joined basically at the center that's known as the centromere, okay? And so we've also mentioned before that DNA consists of genes, and genes are just segments along the DNA, along the chromosome that contains the recipes or the code for making a protein. And across our 46 different chromosomes, we have about 20,000 genes that our cells will use to make different proteins during cellular processes. And so here we have double-stranded DNA up at the top. These blue spheres are histones. And what happens is during right before cell division, the DNA, the chromatin, will condense in prophase. And so this chromatin material is not visible under a microscope during interphase. But whenever prophase happens, the chromatin will condense down, wrap around these histones, and then will condense into a chromosome. One copy of the replicated chromosome is known as a chromatid, right? So this left side is a chromatid, or this upper side is a chromatid, and this bottom side is the other chromatid. And since they're identical to one another because they were replicated, they're known as sister chromatids, okay? And so this whole thing is a chromosome, okay? Now, there are three processes that are super important for DNA to function appropriately, and for it to kind of maintain its function throughout our cells. The first is replication. So DNA needs to replicate prior to cell division so that the appropriate copies can go into the daughter cells. Transcription is the process of the cell reading the DNA and converting that DNA into RNA so that the RNA can be moved out of the nucleus and carries the code of the DNA for a single gene, okay? And after transcription, after that DNA is converted into RNA and that RNA leaves the nucleus, that RNA will then be translated into a protein using a special coded message known as a codon sequence. Okay. So what we're going to talk about first is DNA replication. Okay, so copying DNA before the cell divides. This happens during the S phase, and so this means that the cell is making an exact copy of all 46 chromosomes that exist inside that cell. And the way the replication happens is the DNA will basically unzip and unwind. The two strands will be pulled apart, and then an enzyme will go through and make a complementary strand of DNA that is based upon the parent template strand using specific base pairing rules. And so those base pairing rules are that for DNA, the adenine base pair will always pair with thymine, and guanine will always pair with cytosine. Okay? And the enzyme that replicates the DNA is known as DNA polymerase. Okay? So here we have a figure where at the top you have your parent strand. And the DNA strand here will be unwound and be separated. So you see that this purple strand here is a parent strand, and then this blue strand is the other parent strand. And so what happens then is a DNA polymerase will read each parent strand and will make the associated second strand to that parent strand based off of those base pairs. Right? And so this first parent strand here has an A, or I'm sorry, this parent strand has a T, so the new daughter strand will have an A because A always pairs with T. The second one is a G, so it's paired with a C. The third is a G, so it gets a C. The fourth is an A, so the new strand gets a T, so on and so forth, right? And then what you'll notice is that the other parent strand, right, has the opposite base of the first parent strand. So if the first parent strand has a T for the first nucleotide, then the other parent strand will have an A because they used to be bound together. And so what you end up with is two identical copies, two exact copies of the DNA helix, the DNA molecule, after DNA replication. Okay. Now, mutations can and do alter DNA sequences. So a mutation is just some alteration in the DNA, doesn't matter how it happens, but if it's an alteration and that base pair is not supposed to be there, or if a lot of base pairs are not supposed to be there, or if there are some base pairs that have been removed, then those are known as mutations. Sometimes the mutations can happen during the replication process, and this can happen due to physical or chemical factors or chemical forces. So in our body, we have two different types of cells. We have somatic cells, which are all of the cells in our body except for our gametes, so our sperm and our eggs. So any mutations in these somatic cells can affect the function of the tissues. They can cause cancer. They can, whatever effect that mutation has on that cell will be apparent in that individual, but that mutation cannot be passed on to the descendants because the DNA and the gametes are the only DNA that's passed on to the offspring. So for a mutation to be passed on, it has to happen in the gamete cells. So it has to be in the sperm cells or the egg cells. If it's in any of your other cells in your body, that mutation won't be passed on to your children. And so again, the gametic mutations are what's passed on. Now, our DNA repair mechanism, or I should say our DNA replication process, has repair mechanisms built in. So there are specific enzymes that basically proofread the new DNA sequences and try to look for errors. And if there are errors present, they will cut out those errors, those wrong, incorrect bases, and that section will be replaced with the correct base, the backbone's reconnected, and then the DNA will be fixed, and so that mutation will be removed. So there are a bunch of different DNA repair enzymes, and they're most active between DNA replication, so the S phase, and the beginning of mitosis, right? So basically like the G2 phase. Mutations in certain genes, however, can lead to an increased risk of cancer, and these mutations are mutations that would directly affect like your DNA repair enzymes. So if you have a mutation in a DNA repair enzyme, then that DNA repair enzyme is not going to work properly, so it's not going to catch other mutations, and so that can lead to cancerous cells developing, and we'll talk a lot about how that happens whenever we talk about chapter 18, which is focused primarily on cancer. Now, after so with your DNA, not after replication, because replication happens whenever your cells are getting ready to divide, but the DNA is also part of that transcription and translation process, right? So transcribing DNA to RNA and then translating RNA into proteins, okay? So when we copy DNA of a gene into mRNA, that's the transcription process. And this process happens within the nucleus of the cell. Translation occurs in the cytoplasm at the ribosome. So it could either be the free-floating ribosomes or it could be the ribosomes on the endoplasmic reticulum. And translation converts the mRNA template, that mRNA code, into proteins. So transcription, the way it happens is just like with replication, the DNA will unwind. Instead of a DNA polymerase, you now have an RNA polymerase that will go in and copy those base sequences from the DNA into RNA nucleotides. So the primary transcript, so that primary RNA that's made, includes segments that are known as introns and exons. Okay, introns are these intervening sequences. They basically don't really carry, instructions for making proteins. The exons are the segments of the DNA that are carrying that genetic information. And so the introns are actually edited out of they're edited out of the RNA, and then the exons are spliced together. After those introns have been edited out and those exons have been spliced together, that forms the messenger RNA strand, and then that messenger RNA strand will leave the nucleus of the cell and be ready to be translated into a protein. So here we have a figure of how this happens. So we have that DNA molecule, right? So the purple strands. So that DNA helix will be separated. Your RNA polymerase will come in and it will look at the base pair and then attach the associated RNA base. For RNA, the base pair associations are a little different. So just like with DNA, the G's will always pair with the C's. However, for RNA, the A's pair with U's. So the adenines pair with uracil instead of thymine in RNA. So if there's an A on the DNA base, on the DNA strand, like we see here, the RNA will not have a T, instead it will have a U, okay? If there's a T on the DNA strand, then the RNA will have an A because RNA has the base A. It just doesn't have the base T, so that T is replaced with the U, which is uracil. Okay, so once that RNA, that primary transcript has been made, this is what it would look like. Okay, and what you notice is there are different colors on the backbone. So the light blue are the introns, so this area and this area. The dark blue are the exons. Remember, the introns are those non-coding regions. The exons are the coding regions. And so what will happen is with the primary RNA transcript, these introns will be cut out and removed, and then the remaining exons will be spliced together to form the mRNA, the messenger RNA. Okay, and in this figure showing codons, and so every 3 base pairs in the messenger RNA is known as a codon. I just want to mention that now, and we'll talk much more about that in the next couple slides. So here we have an animation for transcription that goes into this in about as much detail as we just talked about.
RNA polymerase first separates the 2 strands of DNA, and RNA nucleotides present in the nucleus bond with their complementary nucleotides on one of the DNA strands. Remember that G and C bases always bond together, and the A and U bases of RNA always bond to the T and A bases of DNA respectively. RNA polymerase then links the RNA nucleotides together to form mRNA in the process called transcription. RNA polymerase begins at the promoter site and then advances into the gene, separating the DNA double helix at its front end, joining together the RNA nucleotides in the middle, and then rewinding the DNA double helix at the back end. When the gene is entirely copied, the RNA, DNA, and polymerase then dissociate from one another, and the newly formed mRNA is released to go on to the process of translation.
Okay, so that's the process of transcription. So once you have that messenger RNA, it then,
polymerase first.
all right, come on. It then goes on to transcription. And so transcription is why does it say transcription? It should say translate. No. Okay. Anyways, with transcription, right, once that mRNA or once that DNA has been converted into mRNA, there we go. Like we mentioned here, you end up with a sequence that is divided into codons. Okay. And so the genetic code is known as a triplet code, and that's the codons themselves. And so a codon is basically a sequence of 3 messenger RNA bases, and each codon codes for a specific amino acid. And now there's redundancy in the genetic code because out of the 4 possible base pair configurations for 3 codons or for 3 bases, right? That's 64 different possibilities. So 64 different codons, but we only have 20 different amino acids. So that means that there are several codons that code for the same amino acid. And that's important because if you have a mutation, that means that it could still code for the same amino acid and not really cause any changes in the protein. And so several different codons, again, encoding for each amino acid with only one exception. There's only one codon that represents the start codon that starts that translation or the protein making process, and that's AUG. The codon AUG always represents the start codon, and no other codon represents start. So the codon is basically a grammar sequence. Right? So your start codon, like we mentioned, is AUG, and that's at the beginning of every gene. Every protein that's going to be made starts with the codon AUG. Every gene will end with one of 3 codons, which are known as the stop codon. So that's UAA, UAG, or UGA. So this table shows you what each codon basically codes for. So you can see AUG is methionine, which is the start codon. And then here you have your 3 stop codons. And then every other codon represents a specific amino acid. And the way you read this table is left, top, and then right. So let's say we have a codon that is CAU. So what we would do is we would go to the left side first and find the first base, so C. Then we would go to the top and find the second base, which is an A. Then we find where those overlap or where those intersect. So this box. So then we would look in this box for CAU, which is here. And so CAU codes for histine, right? And so let's do another one. Let's do AUC. Okay, so take a minute and find what this codes for. All right, so let's look and see what it codes for. So here's A. We'll go over to U, which is the first box, and then we'll go to C, which is isoleucine, right? So it codes for isoleucine. So let's do one more. Let's do GGA. Okay. All right, so hopefully you found it. So we'll start with G, then we'll go over to the column for G, which is here, and then we'll look for GGA. And so all of the GGX codons, so GGU, GGC, GGA, and GGG, all of those code for glycine. Okay. And what I want you to notice is with this genetic code or with the codons, generally if you change the third codon, you don't really change the amino acid, right? Notice how the, GCX basically, so GC and any other base always codes for alanine. Same thing for GU, so GUX, right, codes for valine. Same thing with CU, same thing for CC, right? You have some cases though where like CA codes for 2, so like CAU and CAC's histine, and CAA and CAG is glutamine. So what this means is that if you have a mutation, right, so like G, A, and then whatever, you're most likely not changing that amino acid, okay? However, if you change the second codon, so like G blank G, right? So you would start with G, and then the second codon tells you to go to a different column. So anytime you change that codon, you're going to go to a different column, and so that's almost always going to change the amino acid. Very rarely do you find the same amino acid in a different column, right? So for example, let's see, I actually don't see the same amino acid in a different column. Now you see the same amino acid, so like leucine, but they're in the same column, right? So they're still UU or CU, right? Now you always get pretty much a change with the first codon because the first codon is going to change your row. So when you're changing rows and columns in this table, you're getting big changes in your amino acids. So the third codon doesn't really lead to that many changes, okay? So that's just talking about mutations and changing those codons at different codon locations, so whether it's first, second, or third. All right, so here's an animation kind of looking at the triplet code, so the codon.
The message that DNA carries is passed on to RNA through base pairing. There are several types of RNA. The form of RNA that carries this message is called mRNA or messenger RNA. Because of the similarities between the two molecules, DNA and RNA can base pair with one new twist. Since thymine is absent in the RNA strand, the adenine base in DNA will base pair with uracil in the RNA strand, U with A. The length of mRNA that results from this pairing is called a transcript, and the process that produces the mRNA strand is called transcription. In the strand of mRNA, each group of 3 consecutive bases is called a codon, hence the name triplet code. Each codon codes for one specific amino acid, and the polypeptide chains that eventually result become proteins after appropriate 3-dimensional folding. This process of producing polypeptide chains and thence proteins using the codons carried by the mRNA is called translation. The genetic code provides the relationship between a 3-base sequence and an amino acid. It is important to understand that the genetic code refers to messenger RNA sequences rather than DNA sequences. There are several characteristics of the triplet code that are important. 1. The code is written in linear form using mRNA bases as letters. 2. Each word is composed of 3 letters. A group of 3 letters is called a codon. 3. The code is unambiguous, meaning that a single 3-base code specifies only a single amino acid. 4. The code is degenerate, meaning that one amino acid may be coded for by more than one codon. 5. The code has start and stop codons that initiate and terminate translation. 6. The code is commaless. The bases are read one after another with no punctuation. 7. The code is non-overlapping. Once translation commences, each base is read in order with no overlap. 8. The code is nearly universal. With only minor exceptions, the same coding dictionary is used by all living things. Marshall Nirenberg and Heinrich Mattei were the first to discover the meaning of one triplet codon, UUU. First, they set up 20 different test tubes, each carrying all 20 amino acids. In each one, they labeled only one amino acid radioactively. In the test tube which correctly determined the codon's meaning, the amino acid phenylalanine was the one that was labeled. Next, the scientists added a strand of synthetically manufactured mRNA composed solely of UUU codons. They also added ribosomes and other molecules in a cell-free system necessary for translation of a polypeptide chain. Now they watched translation occur. They expected a polypeptide chain to form with only one type of amino acid present, since the mRNA contained one codon, UUU. In the test tube where phenylalanine was radioactively labeled, they found a radioactively labeled chain of phenylalanine. The scientists knew then that they had a match. UUU codes for phenylalanine.
All right. So pretty interesting how they found the meaning of the triplet code. I mean, of course, you'd have to do that for each amino acid, but still pretty neat. All right, so translation. Right? So translation is making that protein from translated, or I'm sorry, the transcribed messenger RNA. Okay. So the messenger RNA is the copy of the recipe that is from the DNA. And so the transfer RNA, which is known as tRNA, is a small RNA molecule that basically brings amino acids to the ribosome where translation happens. Ribosomes are made of rRNA, or ribosomal RNA, and proteins. And these ribosomes contain sites for the incoming amino acid and the tRNA molecule. And then they contain enzymes that catalyze bonds between the amino acids. And remember from our organic chemistry lecture that the peptide bonds form between amino acids in a polypeptide. And so the process of translation is initiation, elongation, and termination. So during initiation, you have an initiator, which is tRNA, and it carries the methionine, so the start codon, on the mRNA. And then there's ribosomal subunits that form what's called an initiation complex. So these ribosomal subunits will be a large subunit and a small subunit. Now, once they form that initiation complex, elongation then happens. And so each codon will be read, and the tRNA molecules will bring the specific amino acids that match each codon, elongating the protein chain one amino acid at a time. Termination then happens once a stop codon is read, and that terminates the developing chain of amino acids, and then the protein is released from the ribosome. So here we have a figure of that process. So you have your start codon here, which is AUG. You have your small subunit, your large subunit with the methionine. They bind together and make that initiation complex. Okay. Now, the tRNA molecule has what's known as an anticodon. So this is the opposite base pairs for the codon that it's bringing an amino acid for. So for example, AUG, the opposite sequence of that is UAC, based off of the base pairs. Right? So that tRNA molecule has that anticodon sequence that binds to the mRNA codon. And so if you notice, there are two binding sites in that large subunit. So you have kind of the first tRNA molecule at the first binding site. Then a new tRNA molecule will bring in the amino acid associated with the next codon. Right, so you have CUG codon, then you have the GUG codon. So the tRNA will bring in the one for the GUG, and then the amino acid will bind to the growing polypeptide, and then this tRNA molecule will be kicked out, and then this one with the growing peptide chain will move over into this space, and then the new tRNA molecule will come in. And then once it gets to the stop codon, the completed protein will be released, the large subunit will disconnect from the small subunit, and then the mRNA molecule will be released as well. So here's an animation that kind of shows all of that happening kind of in real time.
mRNA is used in the process of translation or protein synthesis, which occurs on the ribosome. Transfer RNAs or tRNAs carrying amino acids approach the mRNA. tRNAs with anticodons complementary to codons of the mRNA will bind. When two tRNAs carrying amino acids are bound to two codons on the mRNA, the ribosome attaches their amino acids together. The ribosome then moves down the mRNA, exposing the next codon. The tRNA that has lost its amino acid exits the ribosome. This process continues with tRNAs bringing in new amino acids, binding their anticodons to the codons of the mRNA, and having their amino acids added to the growing protein chain. The process stops when the ribosome reaches a stop codon. This codon does not bind with any tRNA. Instead, it signals to the ribosome to stop translation and release the finished protein.
All right, and so that's going to do it for this video. We've talked a lot about the cell cycle and how DNA is very important for the functioning of the cell. We talked about how DNA is replicated to get ready for the cellular division process, and then we looked at the other important functions of DNA in terms of going from DNA to messenger RNA to being translated into a protein. What we're going to talk about in the next videos are mitosis and meiosis, and we'll actually look at mitosis in its own video and meiosis in a separate video as well. And so with that, we'll see you in the next video for this module.